pressure that's allowable will actually go up. That way it doesn't just start bringing heart rate down immediately. So it goes up. So then you would have to dramatically increase physical activity from that point to make pressure go up enough to then want to start bringing it down. So I thought those were some pretty good questions that people had, and feeds right back into the idea that those short-term regulatory mechanisms are short-term for that reason. They reset their baseline. They're not meant to control something like that long-term. So we'll get in chapter 21, we'll get into long-term control through the kidneys, through the renin-angiotensin-aldosterone mechanism, through changing fluid volume and all that stuff. Okay, so we left off then getting into other things that affect heart rate. So not only does pressure affect it, as we saw, and chemical composition, like we talked about the chemoreceptors and hydrogen ion and oxygen, blood oxygen concentration, carbon dioxide concentration. There's also extracellular ions that can affect it. There's temperature that can affect it, heart rate, that is. Temperature is probably pretty straightforward, right? Heart rate increases as temperature goes up, heart rate goes down as temperature goes down. It's also part of the reason why prolonged periods of hypothermia are a little bit more dangerous, is because you're decreasing heart rate along with that. Where it gets interesting is when you start considering a couple of the other extracellular ions. So potassium, I'll try and say this without confusing myself. Potassium levels getting too high is known as hyperkalemia, and the chemical symbol for potassium is K, so that kalemia is going to be tied to that. Hyperkalemia means potassium levels outside the cell, this is extracellular ion, they're a little too high. So think back to what you know about the depolarizing, right, pacemaker potential and all that of your cardiac pacemaker cells and muscle cells. If you've got too much potassium outside, a lot of it's going to start leaking in, despite those slow sodium channels being open. So what's going to happen is essentially you're making your resting membrane potential less negative. And as that happens, it causes issues with opening those sodium ion channels, those slow sodium channels. So what happens overall is heart rate actually decreases with an increased amount of potassium. So hyperkalemia disrupts the repolarizing efforts of those pacemaker cells by making resting membrane potential less negative, and so it then disrupts the subsequent depolarization that happens less often. So heart rate overall tends to go down. The reason I make a big deal about that is because down here, hypokalemia, the other end of the spectrum, little potassium, that also causes heart rate to actually go down. Because in the case of hypokalemia, it's much more polarized or hyperpolarized when we talk about that membrane. So in one case, you're making it less negative and less likely to be polarized, disrupting restoration of that membrane potential. In the other case, you're hyperpolarizing. Not enough potassium ion to offset the positive ion that's being pumped out of the cells. Internal resting membrane potential is lower and lower and lower. That means more sodium channels have to open over a longer period of time to bring it up to the threshold that pacemaker potential. And so cardiac muscle and heart rate heart rate is down. So in either case, hyperkalemia or hypokalemia is likely to result in heart rate. And when you decrease heart rate, you're likely to reduce some stroke volume as well, so longer term effects. You could also get with a decreased pacemaker potential and decreased depolarization, you might even start to get some ectopic accident potential, leading to fibrillation. We mentioned that last time. So that's bad news. You could even get to a heart block, the loss of that action potential conduction through, especially the AV area. So it's important to keep potassium, especially ion concentration, balanced outside of the cell so that you can reset resting membrane, you can polarize the protein, right? So you can polarize on a consistent rhythm. With calcium, hypercalcemia, too much calcium, that tends to lead to an even greater force of contraction because you're able to form more cross bridges, and contract more of that contractile muscle, and because your force of contraction is greater, you're likely also increasing stroke volume to an extent. So it requires that the heart actually beats less, so you may slightly increase heart rate. Increased calcium, hypercalcemia is usually more of a mechanism to get excess calcium out of the blood, out of the hypocalcemia tends to cause increases in heart rate because your force of contraction goes down. You can't form cross bridges. You don't have contractile units going in that cardiac muscle, so the heart has to try to compensate by picking up its rate of polarizing contraction. So whether it's potassium or calcium, you get effects on the heart rate. You could mention sodium in there, but sodium we already kind of know the roles that sodium to try and stay away from too high amount of sodium or too low amount of sodium. So for our focus, we just mentioned potassium and calcium. As far as age effects go, there are some age-related changes. You can mitigate some of these with exercise or regular physical activity. Some of these it's a little bit more difficult. In general, as you age, the left ventricle has to work a little bit harder. Part of the reason for that is maximum heart rate tends to go down some. So especially in the 70s and 80s. So even if they're doing physical activity, it's a little harder for them to keep the heart rate up. So the heart compensates by trying to contract with greater force. And especially if you've got any sort of anything that's increasing aortic pressure or afterload, remember that term afterload. That's the pressure that the left ventricle has to overcome to get blood out of that ventricle and into the aorta. Any of that stuff is going to cause that left ventricle to start working harder, and it's going to hypertrophy the muscle cells and start getting larger and larger. Now, to a point, that works okay. Increased force of contraction, but there is a limit. This is a tipping point. The larger the heart muscle gets in a particular chamber, the less efficient the chamber actually becomes after that tipping point. So hypertrophy of the left ventricle may sound like it does, and it does to a point, but if it's a prolonged thing, it actually makes that chamber less efficient in the amount of blood that comes out of it. There's also less effect from certain hormones, so things like epinephrine, trying to get heart rate up, doesn't work quite as well like it once did. The valves become less flexible, so you can run into recombinant valves, stenotic valves, so those things that we mentioned. Intrinsic conduction system could have some issues, especially with some of those pacemaker cells, so you might see increase in arrhythmias. And then the last two on here we'll cover in more detail. Congestive heart failure, we'll talk about it in a second. Two different types of that. Coronary artery disease we'll cover more so in chapter 21. That has more to do with buildup, including those vessels, plaque, cholesterol plaque, something like that. Or if there's a lot of scarring, inflammatory events, and that could cause issues. So all of those we would consider age-related changes. They're all going to have likely negative effects overall on heart rate because remember the big picture is to get enough blood out with enough oxygen and whatever else to perfuse the tissues adequately and meet the needs of changes in activity, for example. And anything that negatively affects that is going to be bad overall for the individual, right? All right, so from a development standpoint, I like to include this slide in here because it gives some context on when the heart first starts beating, it starts to take on the shape that we're familiar with, and then it brings back in a couple of little structures we mentioned before that the fetal heart help bypass the lungs because you don't need those in play fetal circulation because all of the oxygen, nutrients, and that kind of stuff comes from the blood that's coming from the mother that goes into the center. It's not up to the kids to have the oxygenate its own blood, for example. So just context-wise, I'm not asking you to memorize this, but I like to include this because you can see by the end of the first 3 weeks or so, really the heart is already capable of getting that pumping action. Those cardiac, non-contractile cells, and pacemaker cells are already starting to find their rhythm. About day 24, you start to see polarity with the heart muscles. So you have the arterial end, it's going to take shape towards where the atria are. You've got a venous end that takes shape more towards the apex area of the heart. And so by about the end of the first month, day 28, it's really bent into some distinct chambers. You've got an atrium, you've got a ventricle, the heart's already beating, it's taking blood that's coming from the placenta, and it's moving it from the right side of the heart immediately to the left side of the heart, bypassing the pulmonary circuit back into blood. And then the left side of the heart is pumping that out. And so by about day 35, just over a month of development, you have a true aorta, you have true distinct chambers within that heart, true cava. And for all intents and purposes, you've got a fully functioning right and left side of the heart. What I'll include here, one of the things we've already talked about, but the foramen or valves will open between the two atria. Okay? So both of these things serve as fetal shunts. I want to introduce that term now, because we'll see it again later on as well. But essentially what they're doing with these fetal shunts is they're taking that already oxygenated blood that's coming into the right side of the heart. It doesn't need to go out to the pulmonary circuit, it's already oxygenated. It's giving that blood a chance to mix into the left chambers of the heart and bypass the pulmonary circuit. Foramen or valve allows it to move oxygenated blood from the right atrium to the right ventricle, right atrium to left atrium, and then down to the left ventricle to pump out. And the ductus arteriosus is sort of a secondary shunt that allows anything that stayed in the right side of the heart, the right ventricle is trying to pump out to the lungs by way of that pulmonary trunk. It's a little connection between the pulmonary trunk and the aorta. So whatever's coming out of that right ventricle really just goes into the aorta. So kind of a neat process of how that happens. And then following birth, we kind of mentioned it, the foramen valve closes up, becomes the fossa valis. It happens 10 days or so following birth, because now the newborn can start using its own lungs, its own pulmonary circuit. So both of those things, the foraminal valve and ductus arteriosus, degenerate and close up not long after birth, because now those fetal shunts are no longer necessary. So let's look at some of the representative diseases and disorders of the heart here. Inflammation is always bad news for tissue like the heart. It's bad news for nervous tissue as well. It's bad news for a lot of tissues, especially if it goes on for a long period of time, or if it's really severe. So you've probably heard of endocarditis, you've probably heard of pericarditis, and just different layers of the heart, right? Endocardium versus pericardium, that are becoming inflamed. Different reasons that might happen. Could be an external source, maybe a microorganism or some toxic compound that's accumulating in that tissue, and your immune system, which we'll talk about in chapter 22, goes in and looks for ways to try and get rid of it. It does some collateral damage in the process. Or it could be some autoimmune functions. Sometimes your immune system will attack its own cells based on maybe some aberrant markers that are on your surface. In either case, though, if it leads to any severe scarring of the contractile tissue, or if it leads to, like it did here, stenosis of some of the valve, that's going to have a negative effect on heart function. Right? So endocarditis, pericarditis, try and treat them pretty quick, some sort of anti-inflammatory, microorganism caused, probably some sort of antibiotic to try to reduce the infection continue. In a severe case, and you don't have to focus on this, but I like to mention it, rheumatic heart disease results from a streptococcus toxin following a streptococcus infection. The toxin accumulates in the heart tissue and can cause severe endocarditis, rheumatic endocarditis in that case. This used to be really common back before the advent of antibiotics. So rheumatic heart disease, scarlet fever, those kind of things, very lifetime. And they could mostly be traced back to simple streptococcal infections that just cleared all the way. Okay, so that's inflammation, the inflammation side. On the reduced blood flow side, like I said, we'll mention coronary heart disease, especially in the arteries, in the next chapter. Really, it just comes down to a reduction in blood flow because you've got an occlusion of that vessel. Talk about that process in the next chapter. We've already talked about this idea of coronary thrombosis. Maybe we didn't call it that, but we talked about lung clots forming, right? Thrombus forming. So if it happens in the coronary artery, that's bad news. There's blood flow to the cardiac muscle. If it happened somewhere else and broke free and became an embolism that traveled to the coronary artery, that's also bad. And then the idea of myocardial infarction, heart attack, is essentially just progressive damage to the cardiac muscle cells resulting from a lack of blood flow. So something like a coronary thrombosis could easily be like a myocardial infarction. If you remember from pre-19, cardiac muscle regenerates some, but not real well, right? So if it gets damaged severely enough, and the surrounding muscle can't really compensate for that, that's why we call this progressive heart disease related to MI. That's why we call it progressive, because they don't replace the damaged tissue, and that makes it maybe even more likely that you're going to get less oxygen delivery in the future to not just the heart but other tissues, so it kind of feeds back on itself. And then the congenital heart diseases are failures of development, essentially. There's one or two of these you can see on a couple of slides. Then a septal defect, maintaining that hole, especially if it's the interventricular septum that allows mixing of blood from the left and right sides of the heart back into the heart's pumping effectiveness and the flow of the oxygen content of the blood that's coming out of the left side of the heart. So that can be repaired usually. Sometimes it repairs itself, but we'll leave out the patent ductus arteriosus and patent foraminal valve. If those structures fail to close, then obviously that presents a problem in the result. But we did talk about stenosis of a valve or competent heart valve. Either case, right, you're reducing the efficiency of blood, especially leaving the left side of the heart, carrying the amount of oxygen that it needs to have, all that good stuff. And then we'll talk a little bit more about specific types of heart failure, like congestive heart failure. But one thing to kind of keep in mind, when one side of the heart starts to have issues, it affects the other side as well, because the other side tends to compensate. And it also affects both the pulmonary and the systemic circuit. So there's lots of different things that can lead to heart failure. We mentioned lots of them already. Hypertrophy of the chambers can do it. Even something like hyperthyroidism. Way back we were talking about what thyroid hormone does. It's constantly stimulating metabolism, right, of heart muscle cells. So as they're continuing to do their thing, they're sort of outpacing the ability to regulate heart rate, force of contraction, and all that. And if it goes on for a long period of time, it puts excess strain on the heart, so you can start to see some failure. So there's lots of different things that can lead to that. I'm going to come back to this slide because I want to talk about one specific type of heart failure, the broad category of congestive heart failure, and then sort of walk through why failure of one side of the heart can lead to congestive heart failure on the opposite circuit. So we walk through. Either side can be affected, but let's say it's left-sided failure. So something has happened. Maybe it's myocardial infarction, maybe it's coronary artery disease, but something is causing the left side of the heart, especially the left ventricle, to not be very efficient. When that happens and it injects less blood overall each round of the cardiac cycle, not only is that bad news for the systemic circuit, right, because now you've got less oxygenated blood going out to those tissues, but you've slowed down the flow of blood coming back to the right side of the heart. So it begins to back up now in the peripheral circuit. So left-sided failure, yeah, it's bad for the systemic circuit, but it can also then result in what's called pulmonary congestion, because as we'll see when we get into talking about blood vessels, especially in the little capillaries, the longer a fluid sits in those vessels, the more likely it is that fluid is going to seep out of those vessels, and it's going to start to get into the interstitial spaces. If this is happening in the lung, if you get excess fluid showing up in the lungs, that's a congestion, right? And pulmonary congestion then leads to impaired gas exchange. We'll talk about that in more detail when we get to the respiratory system. But it makes it more difficult to get O2 into the blood, CO2 out of the blood, right? Because essentially what you've done is, get to this in that chapter, but you've thickened the respiratory membrane. So left-sided failure, bad news for the systemic circuit, because now it's being deprived. Also bad news for the pulmonary circuit because now blood's starting to back up in that area and cause some congestion, so blood backs into the lungs. If it's right-sided failure, bad news for the pulmonary circuit directly, right? Because now you've got less chance to load oxygen into the blood and get rid of carbon dioxide. But now, blood is going to spend more time out in the peripheral tissues in the systemic circuit. So this is called peripheral congestion. So you start to see it, especially in the extremities. Feet will start to swell, ankles will start to swell, lower limbs, especially if there's gravity doing some work there as well. So blood tends to pool in those areas, but more dangerously, it'll start to pool in and around the body organs as well, and that can decrease their efficiency to do their normal things. So that's the danger behind that. So congestive just refers to the fact that there's a fluid buildup due to the failure of part of the heart. So if it's left-sided failure, buildup happens in the right circuit or the pulmonary circuit. And if it's right-sided failure, the buildup happens in the circuit connected to the left side of the heart, the systemic circuit. In both cases, it's bad. One failure of one side weakens the other, and you get this idea of decompensation. So a decompensated heart is bad overall. It's not able to get blood out to where it needs to go and keep it moving. So immediate treatment, you got to get rid of the fluid. You can aspirate some of it. You can pull it off of there, and then maybe that helps. But you got to try and get to whatever the underlying cause is. If the underlying cause is maybe chronically high blood pressure that's leading to an increased afterload, like we kind of already mentioned, and maybe you've got to use drugs that can relieve that process, right? That puts less strain on the left side of the heart. If it's having to work harder to overcome that afterload and get blood into the aorta, then that's causing the issue. So if you can remove that strain, then maybe that helps, right? So a lot of people get confused with the idea of congestive heart failure, but both sides are affected when one side starts to have issues. Right? Now, I'll come back to this as kind of our last slide here before we move on to chapter 21. These are three specific congenital heart defects, one of which we've already mentioned, but about 1 in every 500 births leads to a septal defect. And it's really pronounced if it's in the interventricular septum. You can see the little hole here. The key is you're allowing blood to mix. We kind of already talked about that. And so you lose oxygen concentration essentially in the blood that's going out to the systemic circuit. A lot of times this will close up, like I said, with minimal intervention. If it doesn't, though, you can go in and you can graft something over it and you can separate those ventricles once more. A little bit less frequent is something called a coarction of the aorta. If you look at the figure here, you can see there's a severe narrowing just past the aortic semilunar valve. So that goes right towards what we were just talking about. You get a dramatically increased afterload, puts a lot of pressure on the left side of the heart. It increases its workload of that left ventricle to try and overcome that afterload. And again, the more often that happens, and these are congenital, so this is going to happen before birth and then moving on through slightly after birth. The longer this happens, it's going to cause that heart muscle to weaken, hypertrophy first, and then start to weaken. So coarction of the aorta, you have to try and get to that pretty quick and put something in there that's going to open up that narrowed region, especially before maybe the valve becomes stenosed, and thereby reducing the afterload so that you don't put as much strain on the left side of the heart. One of the worst, although it is treatable to an extent, is something called the tetralogy of Fallot. Tetra meaning 4. This is only about 1 in every 2,000 births. But you get some of these defects combined. You get a pulmonary trunk that's too narrow. You've got a coarction there. And that can lead to a stenosing of that pulmonary valve, which then in turn leads to hypertrophy of the right ventricle, increased afterload. It's got to work harder, right? Then couple that with a ventricular septal defect, like we saw in panel A. Couple that with the fact that the aorta opens from both ventricles, essentially. You've got 4 big issues that are drastically reducing pumping efficiency there in the heart, leading to
lot of work done especially trying to work out the specifics of this technology. If y'all are interested in some of the history behind it, and find some stories on that. The struggle in trying to find ways to fix it. But some of these congenital defects are also increased with certain environmental factors. So, especially around the 1950s, certain medications that pregnant mothers were taking weren't really known necessarily to cause a lot of birth defects. They were taking them to address other health concerns for them. A lot of those lead to congenital heart defects and what connection eventually was made. Some of those agents stopped. All right, and the very last thing on here I included, just for context, you don't need to use this for exam study, but it gives you some relationships between like a major issue of the cardiovascular system, which is that myocardial infarction, and the effect that can then have on other systems. Some of them pretty straightforward, like the respiratory system, others not as clear, but these are a lot of the links that have been made into mentions. So just in case you're interested, still might have included that. All right, so that finishes 20. We're not done with cardiovascular system. Here's one of my fun AI generated activities here. I told them to show me the major blood vessels in and around the heart. I think I used, can't remember if I used ChatGPT and NineCafe. But this is what it came with, right? Pretty neat, not anatomically correct, but interesting nonetheless. So we're still in cardiovascular system. We're not done with that. What we've done is we've established the pumping capability of the heart and understand how it works. Setting heart rate, how that can be regulated. What we haven't talked a lot about is what's happening out in the systemic circuit. When the blood leaves that left ventricle, it's going out to perfuse the tissues. We use that term several times. What does that actually do? And how is that actually done? Touched on it just a little bit, and I told you arteries carry blood away from the heart, veins carry blood towards the heart, so we kind of have that idea. But when the blood gets to where it needs to go, there's got to be a mechanism for making the exchange, right, between the tissue and the bloodstream. So we're going to use this chapter. We're going to talk a little bit about that, and we're going to talk some about adjustments that you can make to the blood vessels themselves to, in real time, to direct blood flow or to slow blood flow down so that gases can be exchanged, nutrients can be exchanged. But we'll start pretty broad first. We mentioned these ideas already. We know that pulmonary vessels transport blood away from the right side of the heart to the lungs and then back to the left side of the heart. And we know that our so-called systemic vessels then are doing just the opposite. They're transporting blood away from the left side of the heart out to the various tissues and organs of the body, the systemic circuit, and then they're bringing that blood back to the right side of the heart. So what's important to understand is this is going to use really three different types of vessels. Arteries, veins, and capillaries. And we're going to spend some time talking about the structure of each one of those and why it contributes to the specific function of each of those types of vessels. And then later on, we're going to get into really some of the, I think it's fun stuff. Like I've been talking about flow and the relationship between flow and pressure. We've introduced that idea before, right? Pressure gradients that can affect flow. All of that's going to come into play here, and we're going to superimpose that on top of the idea of blood vessel diameter that can affect flow, peripheral resistance, all that fun stuff. Okay, let's look at the basic functions. Lost some of my numbers here, but we've got 5 that are worth mentioning. Some pretty straightforward. So, for example, circulatory system carries blood to the body tissues and then back to the heart. That one we understand pretty well. What we'll go into some detail on is the exchange methods for getting nutrients, respiratory gases, right, like oxygen, carbon dioxide, getting those exchanged between the bloodstream and the tissues themselves. Then, something we've also mentioned before, transport of hormones, right? Transport
we'll talk about this in chapter 22, different components of the immune system, whether we're talking about white blood cells or inactive complement proteins, inflammatory chemicals, those kind of things. We've already mentioned clotting factors and fibrinogen that are not active, that can become active during the clotting process. And then, of course, enzymes, nutrients, gases, waste products, etc. So the transport mechanism in the blood plasma is going to be the key for that stuff. But we've touched on most of that already. And we've started to touch a little bit on the idea of regulating blood pressure. So we'll see from a blood vessel standpoint how that can be accomplished, in part by changing fluid volume within the blood or by changing peripheral resistance in the vessels themselves, so vasodilation, vasoconstriction, those kind of things. And then very importantly, we'll spend a little bit of time talking about direction of blood flow. So depending on which vessels are wide open and which ones are kind of closed off, you can actually control the degree or volume of flow into or out of certain areas. So this becomes really important as you're moving between a state of rest to a state of fight or flight, for example. We'll see that the cardiovascular system and the blood vessels in particular can be manipulated so that they can direct blood towards, say, skeletal muscles, towards the heart, and away from the urinary system, away from the digestive viscera, those kind of things. So direction of blood flow is going to be a really important aspect, and we'll go into some of the mechanisms behind that. But in order to understand that, we need a little bit of an understanding about the overall structure of these vessels. So unlike the heart, there's no cardiac muscle in a blood vessel. It can still contract to an extent, but it's using smooth muscle to do that. But the vessels, not all the vessels have smooth muscles, so some are able to contract or constrict better than others. So as we go through, what we'll do, working our way towards that, is get a general understanding of the structure of a blood vessel. So think of this as sort of a generalized blood vessel, right? We know there's three big types. I've already mentioned the three main types as part of this closed circulatory system, a closed tube network essentially is what these are. There's arteries, there's capillaries, and there's veins. Right? We've established that before. We kind of said arteries carry blood away from the heart, veins carry blood back to the heart. Capillaries are the areas in the tissues and organs where the exchange of what's in the blood and the tissues actually occurs. So in general, each of these has at least two out of the three layers we're going to mention here, but arteries and veins tend to have all three layers. But these are differentiated tissue layers, and we'll start with the most internal one. So if you're inside the blood vessel, if you're the blood moving through the blood vessel, what you're going to see when you turn and look at the vessel wall is the internal tunic. This is known as the tunica intima, and it's primarily just endothelial tissue. So, and as we'll see, mostly it's stratified squamous epithelial tissue, if you remember the 319 stuff. So it's endothelial tissue. We talked about that in chapter 19 as well, right? When we talked about damage to endothelial cells as part of what kicks off the platelet plug formation and the clotting process. So these are those cells. They're part of the tunica intima. They're connected to the next layer by something called the basement membrane. And that layer nearest the muscular layer, nearest the middle of the vessel, has some elasticity to it, so an internal elastic membrane. Why do you think that's important, considering what the blood vessels are doing, especially the closer to the heart that you are? Why is it important to have some elasticity in these vessels? What is it? So they can expand some, right? So they can do a little stretching, especially if you're really close to the heart. That's a big pressure wave that's coming out of that left ventricle. Remember, we talked about that. And we'll look at the pressure again across the blood vessel network in later slides. But there's a big pressure wave moving through. It's a large volume of blood, especially in those early arteries. And that's going to push on the endothelial layer. It's going to push it out, right? That pressure wave. So if it's completely inflexible, then that's going to cause some serious pressure within that vessel, which could be damaging. But if it's got some elasticity to it, as part of this tunica intima, then it can expand and then it can recoil as that pressure wave passes. And that's going to be a key thing that these vessels, especially those closer to the heart, need to be able to do. And so that tunica intima doesn't have really anything in the way of muscle. It's not capable of doing the actual vasoconstriction or relaxing to do vasodilation. But it does have little openings. We'll come back to these things when we start talking capillaries especially. But these things are called fenestrations. And in the endothelial lining of the vessels that have it, they won't all have these fenestrations, but if they do, these are little spaces where things can move between the endothelial cells of that tunica intima as they're leaving the bloodstream, for example. But they got to have a destination in mind, so we'll see. We'll come back to this idea of fenestrations. The second layer then, the middle layer, is the tunica media, and it's primarily smooth muscle. Right? So it's got contractile capability, but it has to be stimulated. It doesn't really have that pacemaker potential like we saw with cardiac muscle. But these are circularly arranged, smooth muscles that when they contract, they take that tube that they line and they essentially squeeze it down. So in vasoconstriction, the diameter of a blood vessel decreases. Right? And they do it in local areas. You don't necessarily, if you've got a really long vessel, you don't do it along the entire length of the vessel necessarily. You could in sequence, but in general, this type of contraction is going to happen in very specific spots, especially at little branch points, as we'll see later, if you want to redirect blood flow to or away from particular tissues and organs. But that's where the contraction is going to be able to happen, is through this smooth muscle. It's also got some elastic and collagen fibers in this layer as well. But the two big things, again, that are happening here, if the layer constricts, if the smooth muscle contracts, this is known as vasoconstriction. And when that happens and you lessen the diameter of that vessel, blood flow decreases. So if you need to slow blood down to a certain area, or you need to keep it from going into a certain area, you can use vasoconstriction. We'll see some examples of that later. The other side of that is vasodilation. That same smooth muscle, you allow it to relax, blood flow increases to those areas. When you want to perfuse certain tissues, especially if they need a big influx of blood for various reasons, then this is what you would do, the idea of vasodilation. And hopefully you've seen that term before. Y'all remember talking about tissue repair. I think what was it, like chapter 4 maybe in 319. Y'all talked about the inflammatory response, remember, and tissue repair using little white blood cells and fibroblasts. Is that ringing any bells to anybody? All right, you stepped on a nail. I think that was the example they showed. You stepped on a nail and it broke all the way through the epidermis and the dermis and caused all kinds of issues. Well, one of the first things that happened, remember, step 1 was like the inflammatory response. That's made possible because of vasodilation of those local blood vessels. Increase lots of blood flow to that area. That vasodilation also makes it more likely that things are going to move out of the bloodstream and get into those tissues. So trying to make that connection for you there back to 319 without giving you nightmares, right? All right, tunica externa then. That's the externalmost layer. It's a little bit denser. It's a little bit less flexible. It's connective tissue, mostly dense connective tissue. Right nearest the vessel, and then as you move away from the vessel into some of the surrounding tissue, it loosens up a little bit. But those are your three main layers. The thinner tunica intima on the inside, typically with its single layer of epithelial cells. The muscular, smooth muscle layer of the tunica media, doing some vasoconstriction or dilation based on needs. And then the tunica externa that provides some support to that vessel. So it'll be a little different between arteries and veins, and then especially capillaries, but that's the general structure of a vessel. When we go through and talk about these, we'll look at some of the differences. So I included a link to a 3D cross-section of artery, 3D cross-section of a vein with some of these spots labeled so it gives you some context. But one other thing I kind of want to point out here, so you can see the smooth muscle of the tunica media, the endothelium of the intima, and then the dense connective tissue of the externa. why would a blood vessel itself need its own little blood vessels? It's kind of a weird thing, right? Do you think the cells of the endothelium or the tunica media, can't they just get their nutrients and oxygen and all that fun stuff from what's flowing through the vessel? Maybe, but not always, especially if blood's moving through the lumen of that vessel at a pretty quick pace. It's hard to get things delivered where they need to go to the cells of the vessel itself. So the vasa vasorum, we'll come back and talk about those a little bit later as well, they provide an avenue to actually nourish the cells of the blood vessel if the blood's moving fast enough that it can't do that on its own. There's also some nerve support. Notice where the nerves are going. They're going straight to that tunica media because that ought to give you the idea that there's your control mechanism for that vasoconstriction and vasodilation. It's coming by way of those electrical impulses from these nerve fibers, depending, again, on the sensory input that the body's getting. Right? Okay, so here's a quick histology view. You can see an artery on the left here. You can see a typical vein on the right. You can see the lumen, that's the big open space in the middle. You can see a few red blood cells in each of these. But I'll show you this and give you a link to a vessel comparison here, again, just to show you, yes, they do have all three layers, both of them do, but they're not constructed the same way, right? You can see in the artery especially, there's much thicker layers versus some of those that are in the vein, and that's going to come down to part of the function of these things and the amount of pressure that they're being subjected to based on velocity of blood flow and that kind of stuff. So we'll come back to those ideas a little bit later as well. So what we're going to do then is we're going to start doing a rundown of the vessels. We're going to work our way away from the heart. We're going to talk about the arteries first. That's going to then segue into the capillary networks, and then we'll come out the other side into the veins as we work our way back to the heart. Because what we're going to see is not all arteries are going to be the same. There's different categories of arteries. There's different categories of veins. They're going to have a little bit different structural motifs to carry out the functions that they're doing. But in general, arteries carry blood away from the heart, and they tend to get smaller from a diameter standpoint. They get smaller with distance. The aorta, big diameter, right? Nice thick vessel. But as you get further and further away, I think they'll talk about some arteries in the lab this week, maybe, or next week. The further and further away that you get, the diameter of that vessel gets smaller and smaller. Until eventually you get to the capillary networks where the diameters are really small. So we'll see that the larger arteries are the elastic arteries. They're also handling sort of the major pressure waves. So this would be like the aorta and the internal carotids, some of those that are closer to the heart. Then you've got muscular arteries, and these typically are considered conducting arteries because these, or distributing arteries, I should say. Elastic or conducting. Muscular are distributing because they have a more developed muscular layer that can constrict and shunt blood to a tissue or away from a tissue. And then as you head into the capillary networks, arteries are at their smallest, and those are known as arterioles, and they feed into what we'll see as the arterial end of the capillary bed. So, good rule of thumb is that arteries, as they move away from the heart and they get smaller, they'll start to transition structurally, and the smaller they get, they have a less proportion of elastic fibers, and they'll have actually more smooth muscle. So something like the aorta, we'll see, is closer to the heart, much larger in diameter. It doesn't have a lot in the way of smooth muscle. You don't really think of like the aorta constricting, right? It's not really the point of that. You want it to expand and with that pressure wave, and that's why it has elastic fibers. But when we start to talk about, and you will in lab, specific small arteries and arterioles that feed certain organs, now you're going to see they have a lot of control over their own diameter, and they're not handling big pressure waves because they're far enough away from the heart, so they don't need quite as much elastic. They need a lot more smooth muscle in order to do that direction of traffic. We'll start with the elastic arteries here and mention that they're conducting arteries. I don't expect you to memorize the diameter of these things. I just use it for context. The largest is somewhere around 10 or so millimeters if you ever aorta is pretty similar. Most of the named arteries you'll come across, especially when you're looking at lab, these would fall into the category of these elastic arteries. They tend to deal with high pressure. Remember, part of the reason why they've got so much elasticity. High pressure that tends to fluctuate pretty wildly between the peak of that pressure wave in systole and then the trough of it during diastole. So they've got more elastic tissue than smooth muscle, and they've got a relatively thick tunica intima compared to what we'll see with some of the smaller arteries, and a relatively thin tunica externa, and they don't have a really thick tunica media with that smooth muscle. Most